Okay, so today we are going to talk about how things get into and out of the cell. The reason that this is necessary is for the cell to maintain homeostasis. Remember from Unit 1, homeostasis is a characteristic of life. Living things maintain it. And we define homeostasis as an internal balance. Now this internal balance might be temperature. This internal balance might be glucose, making sure that they have energy to process. It might be water, but kind of the things to maintain life and keeping that balance. Now, specifically, we're going to talk about passive transport. When you think of a passive person, it might be quiet, might be reserved, and it kind of has a crossover with what it means with cells. Essentially, we mean no energy is required to do this. So it is a huge saver for the cells that energy is not required to bring things into and out of the cell for the most part. We will be talking about active transport, the opposite of this, which does require energy, but that's for a later time. So passive transport, when we talk about the concentration gradient, passive transport moves either down the gradient or with the gradient. So when we think of high to low concentration, things naturally want to move from a high concentration to a low concentration. So when we say down and with, just think of kind of drawing an arrow down a ramp or in the direction it wants to go. So when you think about dropping a tennis ball down a ramp or riding your bike down a hill, you don't have to pedal. You'll go down the hill without any energy exerted. So when we look at all the dots over here on the right side, there is a bigger concentration up here. There is a smaller concentration down here. So predominantly, we want to move from a high to low concentration. However, molecules do move randomly. We will see a few go in the opposite direction, but predominantly high to low. And then the overall goal is to reach what's called equilibrium. And equilibrium is when we have an equal rate of going kind of in both directions versus a predominant direction. Now we have three types of passive transport. We have simple diffusion, facilitated diffusion, which is essentially simple diffusion but with a helper, and then osmosis, which is focusing on diffusion of just water. And we'll break each one down individually. So simple diffusion, and our example of this was putting the food dye in the beaker and how the food dye spreads out across the beaker over time, because the food dye itself has a greater concentration of dye molecules. The water in the beaker had no concentration, so it had a lower, so we still want to go high to low concentration, and over time equilibrium was met because the dye spread out. So when we think of high to low with these pictures in the lower left-hand corner, we have a higher concentration. On the right side, we will see dominant movement to the left. There will be a little bit of movement going to the right, but predominantly the net movement, overall movement, is to the left. Over here, we have high over here, so we see greater movement to the right. You will see a little bit to the left. As we go down, we see greater movement here. So we're going to see more go to the right, but since that concentration is relatively equal, it shouldn't take very long. A little bit of movement to the left. And since these are equal, I want to draw my arrows the same size, saying that they are going the same rate, back and forth. Similar to what we have over here, there is a high concentration here. There is a low concentration across the cell. So what we see over time is we see a greater amount going across to the low concentration. We'll see a little bit go the opposite direction. But as the concentrations get equal, notice how my arrows get smaller and more similar in size because equilibrium has been met. Equal concentrations on both sides. Now, the second example is facilitated. We're still dealing high to low, which means no energy. Facilitated diffusion is essentially diffusion with help. And that's what we mean by facilitated, is it is getting help going across the membrane. And this help is in the form of what's called a transport protein, or just proteins in general. We have two types. We have channels, which are always open, and we have carrier proteins, which do close and open as needed, and both of these, once again, make up a transport protein. So you may ask yourself, well, why is this necessary? Well, we need to transport large polar molecules like glucose, essential molecule to keep the cell alive, process that down into energy, as well as ions like sodium ion and chloride, which is a chlorine ion. The reason why those need help is because, remember, we have hydrophilic and hydrophobic. And our hydrophobic tails want nothing to do with the charges. Charges go with polar. Hydrophobic is going to be nonpolar, so they need help getting across. Now, the neat thing with your proteins is similar to enzymes, these are going to be specific to one molecule. So you'll have a protein for glucose, you'll have a protein for sodium ion, you'll have a transport protein for chlorine ion. One protein per molecule. Similar to how an enzyme was specific to one substrate. The third and final passive transport is osmosis. And the biggest thing here is just dealing with water. Still moving high to low concentration, still having no energy needed to complete this. A lot of application with osmosis. For instance, the three examples here, why you're supposed to gargle salt water with a sore throat because of the inflammation, putting salt on a slug, watering a plant. If you've ever made the mistake of putting salad that has dressing on it in the refrigerator, over time it becomes very slimy. So we'll be talking about a lot of different things that deal with the osmotic condition. So we have three different solutions that deal with osmosis: hypotonic, isotonic, hyper. And as you can see, hypo is basically swelling in size, iso stays the same size, and hyper, it is shriveling in size. So when we talk about solution, we once again have to break solution down. We have solute and solvent. Remember, water, going back to Unit 2, the universal solvent. What is being dissolved in it is called the solute. Oftentimes we will talk about salt. So the first solution is iso. Now iso simply means the same. Now these three solutions are based off of the solute. But the easiest thing is to simply look at the solute and then kind of compare it to the water solution, or water concentration, shall we say. So when we look down here, we have 10% salt, so that means 90% water. Inside the cell, same thing, 10%, which means 90% water. So since they are both the same concentration, we're going to see equal rates going into and out of the cell. So the shape remains the same. This is ideal for animal cells. Here's your red blood cells right here, kind of staying the same shape. If you've ever needed an IV is not simply just pure water. It is a collection of water and salt to try to mimic the blood concentration to maintain isotonic solution. Our second type is hypo. Anytime I see the O here, it does remind me of swelling getting bigger, because it's easy to confuse hypo and hyper. Hypo means less. These definitions are, once again, based off of solute. So when we look at the solution itself, 10% salt, lower solute concentration outside versus inside the cell, which means we have a greater water concentration outside versus inside. So since there's a greater water concentration outside versus inside, we see much more movement going into the cell versus outside the cell. This causes it to get bigger, it to swell. And this is ideal for plant cells, because remember, plant cells have a special organelle called the central vacuole, which is about 1/3 the size of the cell. It'll swell up with water and create what's called turgor pressure, which it uses for further support. Animal cells, if they get too big, they don't have a cell wall with that extra support. So if they get larger, they're going to burst and die almost like a water balloon. All right, hypertonic. When I look at that E here, especially with lowercase e, it kind of is all shriveled up. Kind of reminds me of what's happening in this situation. So hyper means more. Once again, this means solute outside the cells. In solution, once again, is outside cell. So if we have a greater salt concentration outside the cell, that means we have a lesser water concentration outside. So we go from high to low. So we're going to see a greater concentration of water move out of the cell compared to move in. So if you are moving out of the cell, you're going to shrivel up. So when we see our red blood cells here that are in a hypertonic, this is your body's way of saying, I need water, I'm dehydrated, drink something, in terms of maintaining homeostasis. Now when we look at the grand scheme of things, which of the diagrams best represents the movement in osmosis? Once again, osmosis means water. So we're primarily looking at tracking water. The first example is looking at water molecules moving. That looks good. The second example is simply looking at sugar molecules moving. We don't want sugar molecules. We want water molecules moving. Third one is looking at movement of sugar. We're not focusing on movement of sugar. And then the fourth one, once again, movement of sugar. The only one that is only looking at the movement of water is letter A. So a couple examples of things that we'll be looking at. Celery. We have water, so this would have been the hypo. Very fresh produce. A lot of times supermarkets will spray their produce with water, fill them up, try to keep them fresh, juicy. Also, if they fill up with water, then they weigh more. And a lot of things are you pay them per pound. So you're actually paying more money for water. When you look at the hyper solution over here, it's kind of all shriveled up. It's limp because water was pulled out. So it kind of loses a lot of that structure versus the very supportive hypo. Eggs, you might have seen these in middle school. You dissolve the eggshell. You put something in an isotonic solution like vinegar, which does have more than just water. It is pretty similar to the shape of a regular egg. So this would have been our iso. When we put it in straight tap water, this was our hypo, cause it to swell. It is much bigger, much firmer. And then when we put it into our hyper solution, cause it to shrivel up. You can't really see it by the picture here, but there is kind of a dent right here because it is deflated. Another example, gummy bears. So from day one, we put it in a hypo solution, and it got bigger in comparison. Day two, we put it in a hyper solution, and you can see that this is a little bit smaller than that. It decreased in size because water was lost with what was happening. So, in summation, isotonic, hyper, and hypo, whenever we are looking at solute concentration to figure out our water concentration. So 10% solute means 90% water, 1% solute, 99% water. There is greater concentration in the cell versus outside. We are going to see water leave. This is hyper. Next one, 1% outside, so 99% outside, 90% inside. It is greater outside, so we are going to see water come in, and it's going to swell. Whereas over here, it was going to shrivel. This is hypotonic. Remember that? O. And then finally, we have the same amount. So from a water perspective, 95%, 95%. What this leads to is an equal rate going in and out, and this is iso. Just because we say water goes in versus out, there is still a little bit going the opposite direction. We're looking at kind of the net rate, the overall rate, and that's why I kind of like to draw the iso as two similar sized arrows. Okay, that is it for passive transport. Remember, all three of these are dealing with no energy and going from a high to low concentration. If you have any questions, let me know. Thanks for watching.